Okay.
We can see your screen, Noel.
Oh, good, good. Okay. Can you hear me?
Yes, sir.
Yes.
Thanks. Yeah, we're good.
That's fun watching the numbers go up.
It is.
Who knew we were so popular? For those just joining the webinar, just want to let you know we're watching the attendees continue to join. We appreciate you guys' patience just for a few more minutes, and we'll get things kicked off. All right, I see Noel has his presentation up and ready. We'll go ahead and get things rolling. We'll just cover a couple things real quick. First of all, hello everybody. Welcome to the Mid-Atlantic Biosolids webinar series. We're pleased to have you join us today. My name is Elliot Pomerantz. I'll be your moderator. I'm a member of MABA and excited to be moderating the session today. We've got two great speakers, the first of which is Noel Lyons. He'll be presenting on carbon sequestration potential through biosolids composting, exciting topic. Our second speaker is Aaron Stevens, in place of Trudy, who you may have on your programs. He'll be stepping in on presenting managing the pile, meaning vector attraction reduction, pathogen reduction when process controls are limited in the biosolids process. These two presentations, we'll cap them off after intermissions with Q&A, with a 15-minute technology review on BioRem's product for odor control with Bill Mullin. So before we hand the mic over to Noel, we want to go ahead and have a few housekeeping items just to cover. Those of you who've been on the webinars previous will already have these covered, but just first be aware your microphones are muted. Video has been disabled for the attendees for the moment here. And if you can hear me, you did it right. Next, each presentation will be 25 minutes, roughly. Can be a little bit more or less, but I'll try to keep us close to the timelines in respect of everyone's time. We'll have 5 minutes for questions. Please use the Q&A function and or the chat, and we'll go ahead and try to address those questions back and forth. So let's see. Finally, we'd like to hear from you during the presentation. So again, if you have the questions, go ahead and send them through that function. Today's webinar will also be available after the live session. An email with a link to that recording is going to be sent to everybody in mid-August. So without further ado, we'll kick things off by working with our first speaker, Noel Lyons. Let's see, a little bit about Noel. Noel Lyons is co-founder of McGill Environmental Systems. This was done in 1991. They've been around quite some time. This is 20th year as president. Under Lyons' leadership, McGill has built its business in convincing customers, regulators, that composting can be a serious environmental, economical, and sustainable technology. He's responsible for managing 6 facilities in the US and in Ireland that have composted over 6 million tons and distributed over 4 million cubic yards of products. He holds a degree in agricultural science from Waterford Institute of Technology in Ireland and a certificate of technical competence in composting from the University of Maine Cooperative Extension. He's also presented numerous industry conferences throughout the US, and us at Synagro have had the pleasure of working with Noel as well. So welcome, Noel. We'll go ahead and kick it over to you.
Thanks, Elliot. Thank you very much. Good afternoon, everybody. As Elliot said, I want to talk about carbon sequestration and related to biosolids compost. And put it another way, is maybe the potential of carbon sequestration to be a revenue source for biosolids compost. So today, there are ever-expanding programs and initiatives that address climate change through carbon sequestration. And compost is only at the beginning stages to get noticed. Yet the science, and I'll get on that later, the science is informing us that compost application may actually be the superior practice for carbon sequestration. Today, it's the whole carbon sequestration activity / industry is in the tens of millions of dollars today. I expected it's likely to go to the billions of dollars in the future. So the topics I'm going to cover today are obviously a quick intro on carbon sequestration, which I'm sure many of you understand. Going to talk about McGill calculated its carbon footprint of 3 of its facilities, so I'll do a walkthrough on how we did that. Our next stage is we plan to get that audited. And then I'm going to highlight one key research on compost use and carbon sequestration. Then I'm going to talk about the dollars and cents, what's going on around the country in efforts to monetize it. And finally, I've got some brief conclusions. So get started. So carbon sequestration, again, as many people know, it's the whole act of putting carbon back into the ground and getting the excess away from the air. Typically, when we talk about it, we talk about these 3 types. For the purposes of today, the terrestrial biological is the one that I am focusing my sole attention on. Yeah. So this is a graphic of biological carbon sequestration. And we talk about the short cycle and the long cycle of carbon. Primarily, historically, we've talked about the short cycle when we talk about putting cover crops, putting compost back in the ground and that. And central to the whole thing is my biological activity. We're improving the health of the soil. More things grow in the soil. That means more carbon gets locked up below the ground and indeed above the ground for a short period of time. But ultimately what you're looking for is, in addition to increase in the topsoil layer, you're hoping that looking to have a portion of that carbon become stable, your long-term carbon that's hopefully will stay in the soil a very, very long time. Okay, so where are we? Okay, so I think that, sorry, can I go? Yeah, no, I think I'm right. Yeah, I thought I'd jump the slide. So, how big is the potential? There are lots of research and lots of reports on this, but here's kind of a commonly highlighted one. U.S. rangelands have the potential, it said, to put 330 million metric tons of CO2 back in the soil. But maybe of more relevance to us, it's estimated that U.S. cropland can put something in the region of 770 million tons of carbon back into, or CO2 from the atmosphere back into the soil. Okay. So here's a list of the common practices that are used: planting trees, wetlands restoration, cover crops, no-till farming, and compost application. Probably the no-till and cover crops of the 2 we probably hear about the most. Okay. So I want to talk about compost briefly from a point of view of carbon. Compost, unlike the other practices, compost actually has a double contribution to carbon under certain circumstances. And the first one is the one we talk about more probably. It applies to probably maybe a little under 50%, I think, of the biosolids in the United States is going into landfills. We hear more about this in terms of the other big organic waste source, which is food waste, which much 90-something% of that is going into landfills. So when you divert that away from landfilling, you are contributing, you're reducing the amount of CO2 going up into the air by a very significant amount. And of course, the second one is when you take the compost, the product, and put it out on land, you're sequestering a certain amount of carbon back into that soil. And here's just a quick graphic I like to use to highlight those 2. Now, this was written first for food scraps, but the figures are not going to be a whole lot different for typically dewatered biosolids. So if you divert 1 ton of organic waste away from a landfill, it's the equivalent of taking an average car and driving from here, the Raleigh, North Carolina area where I am, to Utah City, Utah. It's over 2,000 miles. And putting a ton of compost back into the soil is almost gets you from Utah back to Raleigh. It gets you about 90% of the way. So when you look at it from a transportation point of view, diverting from a landfill and using the compost can make, on a unit basis, make a really significant contribution. Okay. So on to McGill's footprint. So we decided last year that we would look back on 2018 and calculate our carbon footprint at the facilities, our carbon, hopefully negative footprint, we hope, expect. So here's how we went about it. We went out to 80% of our customers at all the plants, and we interviewed them individually, and that was 80% by tonnage coming into the facilities. And then whatever conclusions we got from the 80%, we just extended that out to the remaining 20. So the key question we asked every customer was, if McGill did not exist, what would you do with your waste material? We were a bit surprised actually that landfilling came back by far as the highest alternative. Then when we knew where that alternative outlet was, we compared the transportation from the source to that outlet versus to the facility, one of our facilities that the material would be coming to. Sometimes that meant we were closer, sometimes we were further away. So that was a plus and minus exercise along the way. Sorry, wrong button. Then we used it. We use a lot of electricity. We've had aerated static pile technology at all our facilities. So handling electricity is a component of our footprint, and of course diesel fuel for all our loaders. So here are the key assumptions we used. A ton of organic waste, 0.76 tons of CO2, mostly released as methane. Compost processing, the figure that EPA code is 0.18 tons of CO2. We would expect that our figure is measured at our plants. It's probably a little bit better than that. That's taken from a series of composting facilities over time. Ours has, compared to the average, our facilities have quite a lot more process control, including continuous aeration throughout the whole composting process and all through the whole curing process. So when that 0.18 is calculated, it's mostly CO2, small amount of methane and NO2. And that's where I think with the continuous aeration, we would expect that small amount of methane to be less or none. And finally, as we said before, a cubic yard of compost, 700 pounds. Then we use, these all come from the US Energy Information Administration, 4 pounds of CO2 per mile driven, kilowatt electricity. Here we went with the worst case scenario, which is coal. Where two of our facilities are located, most of the energy is nuclear, which would be a bit below the 2.21, but we would rather on the conservative side in the first cut. And then a gallon of diesel fuel, which is basically the mile driven. We use 5.5 miles per gallon, so that 4 pounds is the 22.4 divided by 5.5. So, and very quickly, don't need to dwell on these, but in 2018, the total tons that went into the equation, incoming was 240,000. And the compost made from those three facilities was 208. And then these are just, again, no need to dwell too much on these, I think. Miles, a little under 2.5 million miles. One thing we've been able to achieve is to greatly reduce our loader hours over the years by bigger screening plants, better sized loaders, how we've refined the process. So I think if these loader hours were 10 years ago per ton, I think it would be much higher. So then here is the high level. So at the left and right, you see where the two big gains are, our savings. So the landfill diversion, which is usually the case with organic waste, that's where you get by far the biggest saving. And over on the right is the compost sequestration. So based on this, we calculated our net carbon footprint at minus 204,000 tons approximately. Then I took a look at the figures from another way. So what we did here was, let's assume that zero, if the alternative for disposing of our waste coming into our facilities was not landfilling. For instance, land application. And what we did for the purposes of this exercise is, if a customer told us that the alternative to us was land application, we give that an equal score with composting. So there was no, unlike the landfilling. And we had, I think, one customer who said they might incinerate. We give that, for the sake of the exercise, it was a tiny quantity. We give that the same figure. So if I took all the internal processing costs, the diesel, electrical, and the emissions from the composting process itself, we had a net, also factored in here the delivery of the compost, we had a net carbon, I guess, sequestration of 21,186 tons from those 3 facilities. Some things, I guess, factors we did not consider, which if we were doing a more, I guess, extensive calculation should be. The main one is fertilizer offset. Biosolids compost, I think, as most of all of you know, is a very significant source of nutrients. Our product typically runs somewhere in the region of 2% nitrogen, but also you get P and K and micronutrients. So we did not factor that in at this exercise. On the compost use side, we get information all the time from our customers, that they're reducing chemical use very substantially. And finally, was probably one of the biggest ones for compost that the research is showing us is compost in a way, with the initial application, you're putting carbon back into the soil. But the much bigger prize is the compost is in a way, maybe you could describe it as acting like a stimulant. And it's, there's more long-term research now that's showing that the benefits from the compost are cumulative over a number of years. And some of that has been modeled now for up on 20 years or so. Okay, so briefly on research, there's been an awful lot of research on compost use and that. A lot of it is maybe short-term research, somebody fairly maybe ad hoc, simple enough research. There are a number of places in the country now, though. Mostly, I would say on the west coast, with Marin County and UC Davis, and it's the UC Davis one I want to just highlight. I think Nebra did some information on this. I think Ned Beecher some time ago, which is a really good summary of it. So, sorry, wrong button. Sorry, a moment. Okay, so this is the Russell Ranch, which I think is a 72-acre facility. It's been going since at least 1993, and they have, I think, 72 1-acre plots. And this is the study I want to talk about. And if anybody later wants to see a copy of the whole report, I can easily email that to anybody. And I'm going to talk about just two things in my view that make this study very unique when it comes to compost. And quite frankly, when it comes to carbon sequestration, biological carbon sequestration in general. Issue number one is, we don't have a whole lot of long-term data. So this study is over a 19-year window. So in the composting world, that's truly a lifetime. And number two, which is really, I think, truly game-changing, is the vast majority of research out there gets done in about the top foot, 40 centimeters or so of, at the most, of soil. So it's very unique for to go down 2 meters. They went down 2 meters, and the results are really interesting. So how do they do it? So they use, they have 2 crop rotations that have been going on for a long time. And they have 3 scenarios. The abbreviations, the top one, if you look at the maize area, CONV, conventional farming, synthetic fertilizer. Then they use conventional plus with cover, WCC with cover crops. And then ORG organic. Their definition of organic for the purposes of the study is it's cover crop plus about 4 tons of poultry manure compost. And here are the results. You can see the color bar, the barcode. So from left to right are the depth, and the very right is the whole profile. So quick summary, and I'll give you more detail in the next slide. The conventional and the cover crops both were a net loss of carbon from the soil over the 19 years. But the cover crop plus the compost had a very significant gain over the 19 years. Now, unfortunately, in the past, they have since changed it. They have not done compost on their own. There's a possibility that the combination of compost, the cover crop and compost, was what made the compost work better. I think they expect, and I do, that the major factor there was not the cover crop. It was the differentiating factor was compost. But if you look at it in terms of interpretation, so if this were a sort of a typical study and they'd gone down to say 30 centimeters, that's about 1.25 feet or so, the conventional would be showing a loss of 3.8%, and The real loss is 4.8. The conventional and the cover crop is probably really a huge revelation. So if you would only measure down the 30 centimeters, you would be coming away with the conclusion that the cover crop was increasing the soil carbon. But in fact, when you factor in the whole 2 meters, it's actually not at all. It's quite a significant loss. And the compost and the cover crop, the organic fraction, is the only one that at all levels is showing an increase in soil carbon. And in reference to, there's an international standard, or I guess maybe initiative, called the 4 parts per thousand. Some of you may well have heard of this. Probably the most widely recognized sort of target to drive carbon CO2 from the air down into the soil. And there's been a lot of controversy about whether it's achievable or not. Well, these results actually on the compost plus cover crops actually outpaced what's required to meet the goals of the 4 parts per thousand, or 4 parts per milli, as they call it in Europe. Okay, and then I just want to reference this flying past it. The Marine Carbon Project, and there's a reason you'll see later why I'm referring to this, is kind of the gold standard for large-scale use of compost for soil health and carbon, I guess, carbon sequestration in the country. It's been going on a very long time, and they are doing this, they're doing it research level, but they're also doing this at very full scale, 100-acre plus plots. Okay. So I'd like to make a couple of mentions about the relevance of, obviously they use biosolids, they use, poultry manure compost in this study, but I want to make a couple of points here on the relevance of public conclusions we can draw, or maybe interpretations from that to biosolids compost. And what I want to say on that is, in the world of compost today, I feel as though we have, if anything, we have too many classifications of compost and standards. And in all honesty, many of those standards are not at all based on science, and they're not at all tied to any meaningful analysis. And I think it'd be more informative to look at compost as 2 categories. You really have the high nutrient compost and the low nutrient compost. And they both have their uses, obviously. And I would put poultry manure and biosolids most certainly in the high nutrient compost category. And I think it's fair to say that I would expect biosolids compost, of all the composters, to perform closer to the poultry litter compost, quite frankly, than any other compost that I think is out there. Yeah. Oops. Again. So yeah. And then sorry, yeah, I've made myself getting ahead of my notes here. So the other awfully important one, of course, is we're now learning more and more that it's not just about shoveling carbon back into the soil. Biological carbon sequestration, it's obviously biological activity. Microbes absolutely need nitrogen, and the biggest criticism that's leveled at the 4 part per million initiative is that there may not be enough nitrogen to accommodate that much extra carbon into the soil. So the obvious takeaway there is nitrogen is an absolutely critical component of increasing soil carbon. That balance has got to be right, as indeed other phosphorus and some micronutrients. And I would say poultry manure compost and biosolids compost meet that better than many other composts do. Okay, on to monetizing. So why is carbon sequestration monetized, and how? I guess there are really 3 reasons, as I see it, for why there is a carbon market out there. Number 1, I guess in some jurisdictions, industry has to do that by regulation. So in essence, they're penalized for having excess emissions, and one way to meet the standard is they can buy credits from somebody else. The second area is there are a lot of companies out there who are, in essence, I would describe it as they're penalizing themselves on a voluntary basis, hence the voluntary market. And they're doing it not because, as a general rule, they're doing it on a voluntary basis, but it's not because they sleep better at night. They're doing it because they have determined that the business edge, whether it be public relations, attractive to their employees and customers. They see it as an investment. And the third category is the smaller. There are there are more and more individuals out there who, in essence, penalize themselves for their carbon footprint and basically buy neutrality by buying. You can go online today and buy down to a single credit for a ton. And the average person in the country, I think, has a carbon footprint of about just under 20 tons per year. So you can go on and you can buy neutrality in that regard. And I guess it's something you could boast about at cocktail parties or at your family Christmas get-together, if we will ever again be able to do that. Okay. So the budget. Okay. So how big is the market? The Noble Institute in the Midwest has sized it like this. I took a look at this figure that I referenced earlier, the 770 million. The going rate today is about $15 per ton credit. That would make it an $11.5 billion business. Obviously, all of this would not come from compost. So how does the whole thing work? It's a very complicated thing today, and this is my version of it. You have generators, buyers, verifiers or certifiers, and you've got traders, or they fall somewhere in between. And then off to the right, I put influencers. We're all influencers. If Apple claims to be carbon neutral because of purchasing credits, they're doing it because we're influencing them. So in a way, all of us are and can be influencers. So who are the generators? Farmers, foresters, land managers, composters, and I think in the future, homeowners. It's worth note that turf is now, I think, the biggest crop in America, and there's probably more compost going into the turf market than there is into any, probably other, maybe ag. I guess general ag and turf are the two big markets. And I would expect that zero of that compost today is getting a carbon credit in the turf market. Yeah. So who are these verifiers? There are all these kind of companies who've got various formulas, and really there's a lot of room to improve standardization. Ultimately, verification is down to the buyer. If the buyer thinks your certification method is legit, they're going to buy it from you. And I'm going to mention a couple of companies. I call them traders. I think they like to present themselves as a little bit more. Indigo Ag, who have had a tremendous amount of, I think, publicity. They've raised huge amounts of money, and they've got this grand initiative, the Teraton initiative, and basically they want to put globally, they want to put 1 trillion tons of carbon back into the soil. So they're doing deals with farmers where they have a verification method. It's a third party, and they will pay the farmer $15 per ton of carbon put back in the soil. They will turn around then and sell that ton of carbon for $16.50 per ton. Indigo Ag, who I'm intrigued by, and I'm trying to get to understand them, they're almost like a .com play on it, or they really want to become, in my view, something like the Uber. They visualize having an app in the future, and every activity you have, you can go on the app and pay for your carbon footprint, and they use blockchain to secure those. Buyers, many big companies, and to the right is just any one of us can be a trader, buyer, quite frankly. And then I mentioned just some funds. Cargill, and in the interest of full disclosure, Cargill have been a McGill customer for over 25 years now. But they're investing in a fund, and obviously they have farmers as customers. They're bringing crops from farmers. So they've got this nice closed loop system through a fund where they're paying farmers, and they're paying them by the acre and based on performance. It's in the $30 to $45 per acre. And to my knowledge, compost is not a component of that today. Then on the political front, there are lots of state initiatives going on at the moment. On the political front, there's this Climate Solutions Act, and we're a little bit hopeful about that. It's one of the few things in DC that actually has across the aisle support at the moment, so it may go somewhere. But central to this is paying farmers for increasing soil carbon and soil health. Yeah. Then I want to reference just one interesting one, that's an initiative that's underway in California. It's been going for a number of years now. It's about a $22 million a year grant to farmers and ranchers. They've already, I guess, the grant has gone to 30,700 acres, and they've reduced greenhouse gases with this by an estimated 73,800 tons. But I think maybe of best interest is over half the money. So this past year, over 11 million of the 22 actually was paid out to compost for a compost application. So you can see it was more than of all the options out there, it got more than 50%. So that's really moving the needle. in the right direction. It's making a clear statement that composting is a compost use is a superior option. Okay, very quick conclusions. I believe carbon sequestration will be a multi-billion-dollar industry. It's in the tens of millions today. Compost can be a nice part of that, and I think we need to be paying attention to it. Compost is, I believe, from the UC Davis studies and lots of smaller studies, compost, I think, can rightfully be called the premier carbon sequestrant. We need to get it, and then actually actions needed on that. Biosolids compost should be a component of every municipality's sustainability or climate plan. I think most cities of a reasonable size in the United States today have plans. I do not know how many of them really think about biosolids as a product and a tool that can really, in a very, very meaningful way, contribute to their sustainability. And I think we need a coordinated campaign. I think the MABA, WEF, US Compost Council, we all, I think, need to push the envelope in terms of getting more of these sequestration programs recognizing compost. And finally, over a third of the country population is living on the East Coast. We probably have some of the most degraded soil on the East Coast, but all the major research centers, most of the major research centers on carbon sequestration are on the West Coast. We need something on the East Coast along the lines of the Marin County project. That's it. Thank you.
Okay. Noel, thank you very much for the presentation. That was very, very good stuff. Certainly something we're all anxious to learn more about, and I think you did a great job at presenting the info. We did have one question that I'd like to try to address here at the end of the session. We're just running a couple minutes behind, but
Sure.
Stand by, Noel, and we'll try to get to that here at the end, okay?
That's fine.
All right, if you don't mind, go ahead and stop sharing your screen. We'll kick things over to our next presenter, who's Aaron Stevens. He's going to be presenting on managing the pile. And just a little bit about Aaron. He serves as a project manager for current and pending national clients actively using using Material Manager database. He's responsible for managing conceptual design, development, distribution, and client support of the Material Manager database for managing nutrients from various types of fertilizers. He manages projects involving data analysis and management, technically based solutions, food processing residuals, pilot studies, and provides technical peer review. Aaron actively uses AirModView software, which is graphically Windows-based front end for the EPA AirMod air dispersion modeling framework, and graduated from Pennsylvania State University with a bachelor's and the MS in agriculture and biological engineering. Great resume. So without more delay, let's go ahead and kick it over to you, Aaron. Go ahead and take it from there.
All right, flattery will get you everywhere. So if folks are tuning in now to hear from Trudy Johnston, I am sorry to disappoint, but hopefully it'll be an interesting presentation for many of you. The title, I changed it. I'm calling this the ins and outs of PR/PVAR. I'm talking about pathogen reduction and process vector attraction reduction. And that relates to the topic of today, which is controlling nuisances and magnifying benefits. The plant operation is where you have the most control for the quality of the biosolids. So once it leaves your facility, it is what it is. So you need it to be as good as it can be before it leaves the fence line. There can be challenges in those process controls, and as well as in how we test and manage. So I'm going to talk about some of those thoughts. I'm going to focus today on Class B compliance. So I'll be looking at PR associated with Class B pathogen reduction. Not looking currently for today's presentation at any of the Class A options. First, of course, is making sure that you're using an appropriate PR and process VAR method for the process in place. I'm going to just look at a few processes and talk about the options available. And as I'm sure if you have a biosolids program, you know that regulations dictate that the monitoring frequency is based on your annual dry tons to land application. And it is specific to dry tons to land application. So if you have a biosolids program where only a portion goes to land application, you potentially could have a less frequent monitoring period. However, my recommendation, though, is that you look at your total production of biosolids when choosing a monitoring frequency. That gives you the most flexibility in how you manage your biosolids, and it also is conservative with respect to monitoring and managing the product to make sure it truly is, you know, the highest quality that you're able to produce. Key for the monitoring frequency is also to make sure that you're doing representative sampling. One example of that would be if you're looking at a facility where you're only required to do annual monitoring. For something like fecal coliform, you would want to do your monitoring event in the winter. So that's actually the most challenging time probably for you to meet process control. Calculation methods are something that I'm working with lots of facilities. I have seen some misunderstanding of appropriate methods for doing the calculations. Basically, for process control, I'll see running averages, or more commonly, just looking at daily calculations. So looking at a daily volatile solids reduction, for example. And that is helpful for process control, but for monitoring, for biosolids regulation and compliance, you want to be looking at doing calculations across the entire monitoring period. So looking at volatile solids reduction as an example, you'd be looking at the average volatile solids into your digester during the entire monitoring period, and you'd look at the volatile solids coming out from that process, and those averages then would be the input into your calculation. If we look at aerobic digestion as one of the examples, pathogen reduction is typically met with time temperature, PSRP. That would be a mean cell residence time and temperature between 40 days at 20 Celsius and 60 days at 15 Celsius. So there are fairly long detention times for this process. And with fecal coliform testing is one way that you can do monitoring for pathogen reduction without doing the more extended demonstration of MCRT and temperature. Fecal coliform, of course, is a geometric mean of 7 or more samples over a monitoring period, monitoring window not to exceed 14 days. That geometric mean would need to be less than 2 million NPN per gram or CFU per gram on a dry weight basis. Process vector attraction reduction for aerobic digestion could be volatile solids reduction, but typically is sour, the specific oxygen uptake rate, which is a lab technique for determining that the material is well digested and that given an abundance of oxygen initially, only consumes it at a limited rate, showing that there is not a lot of food available. Alternatives, if you have challenges with sour, which can be an issue with aerobic digestion, especially in the winter, there is an additional digestion bench test, although it tends to give similar results to the sour test. The other alternative or backup that a facility might want to consider is lime stabilization. Again, that would mainly be in the winter. For anaerobic digestion, the pathogen reduction method typically could be either time temperature, PSRP, that's a process to significantly reduce pathogens. The anaerobic time temperature is an MCRT in temperature between 15 days at 35 to 55 Celsius and 60 days at 20 Celsius. And it also, with this pathogen reduction method, you are allowed to look at a linear interpolation between those two points, the 15 and the 60 days. And basically, linear interpolation looking at the 20 to 35 Celsius and 60 to 15 days. Alternatively, it may be easier to do fecal coliform testing of the biosolids that you're producing. And again, as I mentioned before, that would be a geometric mean of at least 7 samples and making sure that it does not exceed 2 million NPN per gram. For anaerobic vector attraction reduction, pretty universally, you're going to be doing volatile solids reduction calculation. I didn't mention before, there are 2 typical methods. The most common is the Van Cleek. It is inaccurate, though, if you have grit retention. And sometimes you might see that where it talks about, if there's a not insignificant amount of grit retention. Of course, what does that mean? The fortunate thing, though, with Van Cleek is that error is it is on the conservative side. That is that if you do have grit retention in your digester, the calculated VSR with the Van Cleek formula will give you a lower calculated number. So you end up being it ends up being a conservative or more safe calculation. The alternative method is the alternative mass balance. And that method does account for grit retention. So if there is considerable grit retention, there may be reason to look at using that calculation. And with anaerobic digestion, again, alternatives would typically be lime stabilization, although anaerobic digestion typically does not see the same seasonal challenges that you would with aerobic digestion. So looking for an alternative is not as important. Lessons learned. I mentioned for the fecal coliform sampling, minimum 7 samples over roughly a 2-week period. You do not want to exceed a 2-week, 14-day window. I did mention already that for smaller facilities, you may want to test in winter. That's when you're more likely to have challenges. One thing I didn't mention is you want to know that you're getting reliable results. And one way of checking for that is to look at the variance within the data. The standard deviation of the log of those individual data points. That's an indicator of the variability, and the result should be less than 0.3. Since we're on a log scale, that's actually telling us that we should be seeing a span less than a factor of 10 in the raw data. Preservation, containers, and holding time are very important. Specific with fecal coliform testing, it by default has an 8-hour holding time. So for the most part, you're looking at getting same-day delivery and start of the test. There is an exception, though. You can do a 24-hour hold time, which is possible for Class B aerobic or anaerobic digestion. However, it's critical that you use EPA method 1680 or 1681. And you would, of course, need to find a laboratory that is accredited by the permitting authority. So you'd want to refer to the permitting state's accredited lab list to look for 1680 or 1681. With anaerobic PSRP, in a multi-digester process, for example, if it is working as expected and we don't have decanting, we can draw a box around that entire system and look at it as if it was one large digester. So the mean cell residence time would work out to be the total volume divided by the flow. So that's, at least conceptually, tells us that the math might be pretty straightforward. But as I said, that's when it's working as expected. So what if one of the digesters was not meeting the temperature to be able to look at a 15-day MCRT? So looking at the ranges, so if you're less than 35 Celsius, you initially would say, oh, well, then we have a 60-day, but recall that linear interpolation is allowed. So if you had if you were halfway between 20 and 35 Celsius, you would then have a MCRT requirement halfway between 60 and 15 days. So what can we do if you have multiple digesters and one of them isn't meeting that average temperature for your monitoring period? You could say, well, let me average the temperature of all of those digesters. And it turns out, though, if you do that, you're not getting an accurate calculation of the PSRP treatment process. There are some other options, though. You could look at dropping the low temperature digester from that total volume. So you could look to see, well, without that lower temperature digester, do I still meet the 15-day MCRT? You could use that lowest temperature digester to calculate the required MCRT. Of course, there's a chance then that if it's low enough that maybe collectively you're not able to meet that now much higher MCRT requirement, or potentially much higher requirement. But another option is to do a calculation of the PSRP per digester, and then you could combine those results. Some lessons learned continued for MCRT. What if one digester is not in service for part of a monitoring period? Or what if volume is variable in one or more of your digesters? What if decanting occurs? Okay. If you have decanting, then total solids must be factored into the calculation. If you recall, when I looked at doing an MCRT calculation of the system, that it didn't factor in total solids, but you do need to factor that in if there is decanting. Over multiple years, grit is going to be building up, and isn't that going to be affecting the volume, the usable volume, I guess would be a way of looking at it, of each of the digesters. So those are all things you might want to consider. How could we estimate system-wide volume loss to grit? I put together an example here. Imagine you have 4 fixed volume digesters, and you do a clean out of each digester on average over 8 years. And when you do a clean out of one, of each digester, that respective digester has roughly 15% of its volume taken up by grit. So how would you estimate how much volume is lost? What reduction would you need to consider in the MCRT calculation to see if you're meeting PSRP? So I put together 2 ways of looking at this. You could look at 1 digester clean per year until all 4 are done, and then you'd wait a gap of 4 years. Or you could say, well, 4 digesters over an 8-year span, I'll clean one every other year. Is there a difference in either approach? Does it make a difference in how much volume you're losing to grit? Turns out there is a difference. So if you use that first approach, where you do a clean out each year for 4 years, and then you have off for 4 years where you don't do any clean outs, at the end of that 5th year in the chart I'm presenting here, that is where the worst case grit, accumulation grit exists. And across all 4 digesters, it averages 12.19%. So it's less than the 15% at least, but it is fairly high. You're losing 12% of your capacity on average over the years, and that then makes it more difficult to meet the MCRT requirements. I derived an equation. It's up on the screen, up in the blue box. And that equation will take the inputs that are plotted or provided A, B, and C on the left-hand side of the screen. The other method I hypothesized is, well, let's just do one every other year. So everything else is the same, but the worst year is, in this case, it's in year 2, but it's actually the same answer in year 4, year 6, year 8. A loss of, on average, 9.38% of the volume. And there's a different equation for solving that without having to build this matrix. And of course, it turns out you don't need to know what your cycle length is. So the 8-year cycle, the B term, does not appear in the equation in that blue box. So I just wanted to show some things you might want to think about when you're looking at grit buildup. So it affects your volume, which affects the PSRP calculation. It also will potentially affect your volatile solids reduction calculations. So as I previously discussed, Van Cleek is most common. It is inaccurate if there's grit buildup, but it is conservative. I recently ran across a case, though, where the Van Cleek equation was not conservative. If you are in a scenario where you have grit that's being produced, so the grit, you're getting more grit out of your digester than what you're putting in, it turns out that you get a non-conservative calculation. The way of getting around that would be doing alternative mass balance. Now, I worked up 2 scenarios here to share with you, and volatile solids in is on the top, volatile solids out is on the left. And in these scenarios, I banded off here on the top edge from 76 to 86% volatile solids to say, well, typically, you might expect your volatile solids to average somewhere in there for volatile solids in. And if that's the case, we can look at volatile solids out, and I can guarantee you that, well, if you have volatile solids out of 80% or more, you're going to fail volatile solids reduction, guaranteed. And if you're in the yellow area, it's possible to pass or fail, depending on where you are. But if you are able to produce biosolids that have a volatile solids of 66%, you're guaranteed to meet volatile solids reduction. This is using Van Cleek. So we're seeing a sweet spot of, if you at least get down to 66% in this hypothetical example, you're going to produce very high quality biosolids. If we look at alternative mass balance, if there is a fixed solid loss of 2%, a modest amount, it doesn't change the calculation. But if you look at a larger fixed solid loss or buildup in the digester, 10%, instead of the sweet spot being 66, alternative mass balance would actually allow you to pass volatile solids reduction calculations if your biosolids, volatile solids were as high as 68%. So again, it's not the best approach at doing this calculation, because we're inputting a potential range. We're not looking at a specific average for your monitoring period, but I think this does elucidate that the volatile solids out, of course, is critical for making a quality biosolids. I have some notes here on sour. I'm not sure where I am on time, though. I suspect I'm coming up toward the end.
Yeah, just about 2 minutes here, Aaron.
Okay. So with sour, this is, of course, for aerobic digestion. Time of year can be a real challenge. There aren't a lot of things you can do once you have a system in place, but if you have the opportunity to plan ahead, you can have your tanks insulated. Below ground tanks will perform better in the winter. Or if you want to continue a biosolids program, you may need to have a secondary vector attraction reduction option available. Some folks, though, what they'll do is go to a facility that provides its own vector attraction reduction, such as mine reclamation. So they're doing incorporation of the biosolids at a mine reclamation site. So under their mining permit, that's where the VAR then is being applied. In aerobic digestion, you may have insufficient DO. It could be due to poor mixing, which could be an issue from the material being too thick in the digester. Could be an issue with the spargers that are putting air out. You may not have enough air. There are lots of things that can affect this. You could be overloaded. So you might want additional tankage, or if you're bringing in whole waste, you may want to consider throttling back on that. With insufficient MCRT, basically things are running through the plant too quickly, through the digester too quickly. So improving your thickening is a good way of addressing that. Additional tankage, or looking at the loading going through the digesters. With the sour test itself, make sure that you're getting a uniform linear drop through that test. The best way to do that is make sure that your initial oxygen is sufficiently high. You want to make sure that it's well agitated. So you're going to be looking at 6 to 8 milligram per liter of oxygen to begin the test. And when you're doing the test, if the oxygen drops below 1 milligram per liter, stop doing the test, because the microbes are going to be oxygen limited. You're no longer going to see a linear decrease in the data, which is then outside the scope of the test. Of course, there's a temperature correction requirement, and there is a statistical test that's appropriate. In Pennsylvania, that is pretty much done automatically through a state-provided spreadsheet. In your own region, state, you'll want to see if a similar tool is provided. If not, look at adopting something that will apply temperature correction and statistical testing of the results. Thank you very much. If there are questions, I can stick around at the end of the session and do my best to answer them. Thank you.
I'm that muted guy. Yeah, so thanks, Aaron. I appreciate you going through all that. Very, very comprehensive information. We did have one question. I'll go ahead and see if we have some time at the end. If we don't, for everyone on the call, we'll be sure that we field the questions out to the speakers and get them out to those who attended the webinar today. Our last speaker here today, Bill Mullin, will be handling our technical presentation on BioRem. So go ahead and take it away, Bill. Oh, Bill, you're on mute.
All right.
There we go. Beautiful.
Okay. Well, thanks very much, everyone. I'm going to talk about controlling odors from biosolids and organics, because we've seen a number of presentations in this series talking about co-digestion. So we'll include that in our presentation, because once you start taking organics in a wastewater treatment plant, it does add a few other odor sources. Okay, so some quick learning objectives. Really just wanted to talk or highlight that there really is an increased sensitivity around odors, especially around any new facility. Especially, if there's a municipal agency involved, town councilor or a mayor, they don't want to get a call about an odor problem. The second thing that we want to highlight is odors are really a mixture of contaminants. Often in wastewater treatment plants, we've focused on hydrogen sulfide. But when we get into biosolids and organics, there's a whole bunch of other compounds we have to consider. And so we need to match the treatment technology to the contaminants generating the odors. A bit about us. We're probably the oldest company into biological odor control in North America. Been around since about 91. Have about 1,400 installations, about 1,000 in North America. And a lot of the work we do is with municipal or public agencies, either in WRFs or in organics treatment, food waste treatment. And I think what sets us apart is we do have a specialized engineered inorganic media that's formulated for controlling odors from biosolids and organics. So just quickly, some big challenges obviously now in municipal odor control is people are moving closer and closer to a treatment facility. And that obviously creates a lot of challenges. You can't say, well, we were here first. That doesn't really matter to a homeowner. I guess the second thing is the whole quality of life is becoming more important for stakeholders, things like odor, noise, and truck traffic. We found in a lot of these new facilities that are being cited, the neighbors are certainly more concerned about odor than they are about greenhouse gas reduction. So they think the project's great, they just don't have any troubles with odors. And again, historically, we've looked at hydrogen sulfide removal in wastewater treatment plants, but when we get into biosolids and organics, there's other compounds like organic sulfur compounds, DMS, and VOCs that have to be considered. And so a technology that was designed to treat H2S won't be as great just working on those other compounds. Here's what I'm trying to highlight here is if we look at the scenario one, that would be maybe a typical wastewater treatment headworks or something where most of the odor is coming from hydrogen sulfide, maybe a little bit from organic sulfur compounds like DMS. So when we're working with a typical hydrogen sulfide removal technology like a biotrickling filter or a chemical scrubber, you'll actually get pretty good total odor removal. It might not work so great on some of these other organic like DMS, but you'll still, because it's such a small fraction, you'll still get great odor removal from a system like this. But if you go downstream into biosolids processing, organics, where we have a much higher concentration of organic sulfur compounds or VOCs, and if we don't get great removal of those compounds, we're only going to get about 50% total odor removal. So it's important when we're again looking at biosolids and organics that we pick a technology that will treat all of these compounds for us. So here's a quick picture of what you may see when you start getting into organics, and that's a tip floor. I mean, obviously in a wastewater treatment plant, we don't produce things like this. So they get hauled in, often in a very tall building, dumped on the floor. This is from the city of Toronto. It's actually not a co-digestion, it's an anaerobic digestion facility. It's just doing food waste, but it comes in bags. So we've got this big tip floor, probably 40,000 CFM of air going through there. So it's a lot of odorous air we have to deal with. And then there's another room that has depackaging. So this equipment tries to remove the plastic bags and the plastics and separate out the organics before it gets into the digester. So we've added a whole new set of odor sources to the wastewater treatment plant. And again, these are very low in H2S, higher in VOCs and organic sulfur compounds. So just quickly on odors, I think the US EPA a few years ago said 78% of the complaints they get are about odors in their air emission group, but they actually don't even regulate them. But that's how important it is to the stakeholders, because they smell it in their backyard and it bothers them. And generally, a lot of the odors from these facilities are really below the level of something that's going to cause any health concern. But the concern is, again, it psychologically bothers people. It's stressful to smell these odors. So these are why odors are becoming more important to manage. One of the conventional technologies for dealing with odors was a wood chip biofilter. And this is actually one we put in over 20 years ago with the city of Toronto. And what you're seeing is weeds growing in the wood chips as they compost. They break down after a year or two, so you don't get great air distribution. Generally, they have a very large footprint. And once that wood chips degrade and become compost, you have a higher back pressure through there, so larger energy, and then just generally a lot of maintenance and poor odor removal. So it's a low capital cost, but very expensive to maintain because you might have to change those wood chips once or twice a year. So this wasn't working for a lot of facilities. So we came up with an engineered inorganic media. So the core of the media is inorganic, so it never degrades. And then we have a coating on it that's specially designed to remove those compounds generated by biosolids and organic waste. Here's a little idea of the way the system works. It will take a certain amount of water on the particle. So it will take the compounds like H2S that are easy to solubilize and get them out, but also take some of the tougher VOCs and adsorb them. So there's really two things happening with this media. One is that solubilization, so it gets compounds out easily like H2S, plus some of the tougher compounds that aren't so water soluble, it adsorbs out. So it gets both of them out of the airstream, and that's why you get much better removal. And again, it doesn't degrade, so you get long-term reliable performance, and it's engineered as an odor control system. That's technology. That's what it was made as, whereas wood chips tend to be just a waste product. The other thing is there's a very low background odor on it. When you're doing odor testing, even the odor of the composting wood chips, it gets detected. So if you have a lower background odor, you can get better odor removal. So there's just a quick picture of what the media looks like. It comes in varying sizes. You can see a coating on there that has the absorptive capacity, and there's lots of open space to get the air through. And here's a kind of a quick schematic of how the system works. The foul air comes in, Goes through a humidifier here because we want to try to get the air as moist as possible so it doesn't dry out the media. Comes through a distribution grating, slowly makes its way up through the biofilter media here, where the compounds are adsorbed and solubilized out of the air and degraded, and then the clean air is exhausted up here. Here's a quick picture of an installation in Florida. Again, the humidifier is here and the biofilter media is here. This one is constructed of concrete. This is an in-ground system. Again, the humidifier is here and the biofilter media sits below the grade. Also have smaller modular units. These are good to about 5,000 CFM. So in smaller systems, it's easier to maybe make it out of stainless steel or FRP. And this is a bigger system that we had at a food waste composting plant in Toronto. Again, made of concrete, 5 or 6 different cells. And that's it. Back to you.
Excellent. Thank you, Bill. Great presentation. Really appreciate you going through that. Certainly, it's a topic that is very in the forefront of everyone's mind in this current marketplace. So that's great that we're able to see another technological advance in how to handle this. I'd like to take the last couple minutes. We do have a few, and in the interest of going, starting at the top, just 4 questions. First one is for Noel, if you're still on board. The question comes from the slide that you had that said factors not considered. And the question was, what chemicals are reduced in that particular one?
These are chemicals that are used in obviously in mostly herbicides. So we have a lot of product that goes into the golf course industry. And the feedback we get is they're by-compost really to build up the soil health, and then what they're finding is that there's very substantial reduction in their chemical use.
Okay, all right, great. Hopefully that addresses the question. If not, go ahead and please reply on the Q&A. The next 2 questions we had were for Aaron. If you want to queue up your video there again, and the first one was, have you looked at mesophilic temperature-controlled aerobic digestion? This technology operates at 95 Fahrenheit throughout the entire year.
Yeah, in what I was presenting, I didn't look specifically at the various variations and combinations of aerobic, anaerobic, heated systems. But yes, that having a system that is operating year-round at a warmer temperature would avoid some of the challenges in the traditional aerobic digester.
Gotcha. So it makes me think about it a little bit. So would the calculations to meet your pathogen reduction, vector attraction reduction be the same as the other aerobic digestion technology for PSRP?
If it is being considered an aerobic process, you would be looking typically, yes, at the same PR and process VAR. Depending on what you're doing, though, some of them like ATAD could even go into Class A PR. So then there would be some differences.
Gotcha. Okay, great. And then the other question was, can you please send calculations for digester cleaning?
We're probably referring to the equations that were derived, looking at the different scenarios of how cleanouts might be operated. Yeah, I'm happy to share those. I mean, they're in the slides, and I understand that this webinar is available as a recording. But yes, I can also provide some further information.
That'd be great. Yeah, Aaron, if you wanted to shoot that over and maybe to Bill and Kathy and myself, we'll make sure it gets out to the person who answers the question. All right, great. Thank you. That's all the questions we had for Aaron and one for Bill, and we'll go ahead and wrap up. How do you, in your research, calculate or quantify odor detection?
Well, there is actually an ASTM method for collecting odors. They collect a bag sample, believe it or not, and take it back to a lab with a panel of people who have sort of trained noses, and they, send it to about 8 people and average the results out. So there is a method for it.
Gotcha. But this is people testing, not instrumental testing. Is that what I'm hearing?
Yeah, I don't think there's yet an instrument. There's instruments that will proxy measure some of the compounds. They call it an e-nose, but I don't think it's really anything but the human nose that they really use it for reliable assessment.
Gotcha. Well, maybe there's another profession in the works for all of us there. So thank you, Bill. That's all the questions we had for the webinar today. Just want to thank everyone for a great Q&A session. Thank our speakers today, Bill, Noel, Aaron. Thank you for joining, and Aaron certainly for stepping in for Trudy. And I think we're pretty much wrapped up. Kathy, did you want to just share